Assalamualaikum and hi. I'm Dr. Siti Khadijah Adam and our topic today is lipid lowering agents, the drugs that are used for hyperlipidemia. So at the end of this topic, I hope that all students will be able to recall back what you have learned in the previous semester about the metabolism of lipoprotein and also cholesterol transport. Also, you are able to identify some of lipoprotein disorders, and you are able to describe and give examples the lipid lowering drugs. And in this topic, I will explain on the 5 types of lipid lowering drugs such as HMG-CoA reductase inhibitors, fibrates, bile acid sequestrants, ezetimibe, and niacin. So you have to know the examples of all these drugs, their mechanism of action, the side effects, and also a bit of their clinical uses of these drugs. So, what is cholesterol? Is cholesterol important to us? Cholesterol is actually the essential component of our cell membranes and also serves as a precursor for synthesis of steroid hormones, bile acid, and vitamin D. Cholesterol comes from 2 sources. We can get cholesterol from our diet, and also we can synthesize cholesterol in the liver. Our liver makes about 75% of cholesterol, and the other 25% usually comes from the food that we eat. So what happened? The cholesterol from our diet. When we eat any food that contains fat and cholesterol, our enzyme lipase will convert the fat into smaller molecules or emulsion droplets, which will combine with bile salts to form micelles. Mile cells are constantly breaking down and reforming to form monoglycerides and fatty acids. These free monoglycerides and fatty acids manage to enter enterocytes by diffusion. Once in the enterocyte, monoglycerides and fatty acids are resynthesized into triglycerides or TG. TG is then packaged with fat-soluble vitamins into chylomicrons to be transported into the circulation via the lacteals. Now, for the synthesis of cholesterol in the liver, it only happens when there is a low intake of cholesterol from our diet. So reduced intake of dietary cholesterol causes an endogenous production of cholesterol in the liver. The synthesis of cholesterol starts when acetyl-CoA is converted into HMG-CoA. The rate-limiting enzyme, which is HMG-CoA reductase, will then convert HMG-CoA to mevalonic acid or mevalonate. And finally, cholesterol is formed together with TG, which comes from our diet. They are transported from the liver as VLDL into the circulation. VLDL is very low density lipoprotein. Now, the dark blue here indicates the cholesterol ester content in the VLDL, whereas the bigger portion or the light blue color here indicates the content of triglyceride. So that's why it is called as very low density lipoprotein, because it only contains a very low density of cholesterol ester. In the circulation, VLDL will release some of its triglyceride content and free fatty acid by lipoprotein lipase to be stored in adipose tissue. This will form VLDL remnant or intermediate density lipoprotein, which is smaller than VLDL. As you can see, the size is smaller, and also the light blue color, which indicates triglyceride, is smaller than VLDL. VLDL remnants are converted to LDL by further loss of triglyceride. So you can see LDL only contains cholesterol esters. LDL provides the source of cholesterol for incorporation into cell membranes and the key to atherogenesis. LDL will be taken up by LDL receptors in the liver and peripheral tissues. In the liver, LDL will be recycled to form a new cholesterol molecule. Cholesterol from tissues can return back to plasma by being transported by HDL. They are brought back to the liver for disposal, and this is called as reverse cholesterol transport. So, as I mentioned before, LDL will provide the source of cholesterol for incorporation into cell membranes and other body tissues. And that is why LDL is called as bad cholesterol, whereas HDL transport cholesterol from tissues back to plasma and then back to the liver for disposal. So that's why we call HDL as a good cholesterol. Okay, so this is just a table that shows the different types of lipoproteins and also the lipid that contain in each lipoproteins. Okay, so as you can see here in the table, there are 4 main classifications of these lipidemias. We have hypercholesterolemia. Remember, cholesterol is high in LDL. So usually hypercholesterolemia is related to the high level of LDL cholesterol, or hypertriglyceridemia, hyper-TG. TG is highly contained in the VLDL. So that's why hypertriglyceridemia is usually manifested by high level of VLDL. We can also have mixed hyperlipidemia. So it's a mixture of LDL and also VLDL. Or we can also have low HDL cholesterol, the good cholesterol. Okay? And we also need to know the different type of primary hyperlipoproteinemias. For example, primary chylomicronemia is manifested by a high level of chylomicrons and VLDL. Other examples such as familial hypercholesterolemia is a genetic condition. It can be heterozygous or homozygous, and both of them are manifested by LDL increase. So to manage the different types of hyperlipoproteinemias, we need lipid lowering agents. So the 5 lipid lowering agents that I will mention in this lecture is, the first one, the most commonly used, is HMG-CoA reductase inhibitors. The examples are lovastatin, atorvastatin, simvastatin. As you can see, all these drugs have statin at the end of their names. So they are usually called as statins. The second group is the fibrates or lipoprotein lipase activators. The examples are the fibrates, for example, clofibrate, fenofibrate, and also gemfibrozil. And we also have resins or bile acid sequestrants. The examples are cholestyramine, cholestipol, and also cholecebulum. The next is niacin or nicotinic acid. It is a lipolysis inhibitor. And the last one is ezetimibe, which is the cholesterol absorption inhibitor. So you need to know the mechanism of action of all these drugs, the side effects, and the clinical uses of these drugs in managing hyperlipidemia.